like to begin by welcoming you to what we're calling the Grad Life podcast presented by GSA. And so, first of all, my name is Edgar Perez Lopez. I'm the Documented Affairs Officer for GSA, and I wanted to welcome you to our little podcast. So, first of all, I wanted to ask you to tell us a little bit about yourself and your research. Where did you attend school, your research focus, and current goals in your research lab?
Well, before I answer that question, I want to thank you, Edgar, for inviting me to be part of this series of interviews, be part of your podcast. And yeah, I'm gonna try to share some of my experience today with you and answering your question particularly. I grew up in Mexico City, and I attended the National Autonomous University of Mexico, or the UNAM, where I obtained a degree in mechanical engineering. And the research that I'm currently conducting is for many reasons, but one of the reasons I had the opportunity to perform a one-year research outreach experience at the Eidgenössische Technische Hochschule, that's in Switzerland. And I had a narrow perspective of what a mechanical engineer could do. And I discovered there that mechanical engineering can be applied to other fields outside of the classic mechanical engineering fields, and biology was one of these fields, investigating how mechanical forces or physical parameters regulate and affect biology and biological function. So I found that quite exciting, and decided to open a new path based on that experience. Yeah, I hope I answer. I can continue going on.
Well, I guess the next question is in regards to that. What kind of biological training did you have to undertake to implement mechanical engineering aspects into biology? So what did you have to learn in biology to say, okay, I'm going to do research on this?
Well, that is still an ongoing activity. I still continue to learn biology, and it's really pretty much by reading. Like, of course, the vocabulary, the jargon, is very different between each discipline, and the only way to educate one yourself is by reading and discussing and chatting with, approaching, for example, biologists. We collaborate with biologists to make sure that actually what we're doing makes sense. Reading and collaborating and talking with people, with colleagues.
Right, and I guess to touch up on that specifically, on your website, you mentioned research topics such as biotribology, bioadhesion, epithelial, mechanobiology. Can you briefly give an explanation of each and the purpose of these research in these areas, the research that you conduct in these areas?
Yeah, so I've mentioned that I became, to some extent, obsessed or very intrigued and interested in applying my mechanical engineering knowledge to biology. And yes, we need to learn biology and collaborate and talk with biologists, but that doesn't mean that, or let me rephrase that, it means also that we can be inspired by nature and then learn from nature. Not only biology is very broad, but what biotribology, bioadhesion means is that we are interested in understanding how some biological systems are capable of solving challenges that we as scientists or engineers have been struggling to solve. Let's say for the bioadhesion thrust or theme that we are pursuing. You most likely, and the listeners are most likely familiar with the difficulty of gluing two bodies or two components if they're wet or if they're dirty. But marine mussels, or in general many other marine organisms, have developed mechanisms to overcome that challenge of adhering to wet surfaces with very extreme conditions, with high ionic strength, high tides, shear forces. So how are these marine organisms mediating that? That's something that with collaborators, of course, are trying to understand. And we can learn some design rules, molecular architectures, synergistic interactions between chemical moieties that we can maybe inform others, people that are doing synthesis, to come up with clever, synthetic, novel adhesives. And biotribology is the same. We are interested in understanding frictional lubrication of the most powerful and robust tribological system that we can find. And it's our joints, our synovial joints, like elbows, knees, for example. Again, as engineers, as tribologists, we have not been able to come up with lubricants and surfaces that lubricate with such low friction coefficients and for so many years. If you and I remain healthy for 80 years, that means our joints are going to operate efficiently for hopefully 80, 90, maybe 100 years. No, in mechanical systems, we need to change lubricants regularly. We need to change the components also after a certain amount of use. So how is nature being able to develop or to design such systems? And mechanobiology, epithelial mechanobiology, this is also how applying my mechanical engineering background. So at the end, we have cells interacting with others to provide particular functions. So how are these cells organizing and interacting with other cells, with the substrates, to provide, for example, control the permeability of the layers that they form, the epithelial cells.
So the one thing that you mentioned right now was organisms that live in the ocean, and one thing that was coming to mind was there's some extreme creatures, organisms that, for example, can produce a large amount of force within this ocean. And so does your research or any research in these specific subjects simulate the environment that these creatures are in as well? Like, does the environment that we see this occurring at affect like bioadhesion? So for example, if we have a jellyfish in the ocean and we know that it has adhesives on its tentacles, right, or it has some sort of properties in the ocean, and those properties differ in land. So my question is, like, do you look into the environment as well as the organism or the biological aspects of something like an organism?
Yeah, well, at the end of the day, also what we want to do is to control the environment or the conditions under which we can apply the molecular constructs that we will be designing. No, let's say we take some of the molecules, some of the proteins in some of these marine organisms, and of course, they have been designed by nature to operate efficiently under certain conditions. Let's understand how those conditions affect the performance. And of course, we can start to play with the conditions, change temperature, change the solvent, change the ionic strength, and understand how are we affecting the performance. And by understanding how we affect the performance, we can then start to tune the molecules or the proteins themselves to make them perform efficiently under, let's say, physiological conditions, if we want to use these design rules to develop bioadhesives that we can use to replace solvers, for example. Does that make sense?
Yeah. So I guess for the audience, at what scale does your work, in terms of size wise in meters or whatever as the unit you want to use, at what scale does your work generally take place? Are you working within like the nanometer scale, only with cells, or do you scale up a little bit to study other biological aspects using engineering?
That is a very good question, Edgar. So it depends on the project itself. For biotribology, the questions that we are asking are focusing at very small length scales, the molecular scale, and the same applies for biotribology. We work with they can be large or small molecules. We're talking about nanometers. So this is 1 times to the 1 to the minus 9th meters. Now we can change the length scale depending of the system that we're working at. So this length scale that I'm mentioning is for molecularly thin films, but we work with, there's something called the mesoscale, in which one length scale, so at the end we work with we are living in a three-dimensional space. So one of the length scales that we investigate is very, very small, so at the nanometer scale. But we cover these large surfaces, macroscopic surfaces. So these are millimeters or centimeters, about the size of our thumb. So we cover surfaces that are macroscopic, really, but with extremely thin films. And then we investigate how these macroscopic surfaces with very thin films behave under the controlled environment. So for the epithelial mechanobiologists, and that is the research that explores how mechanical forces affect and regulate cellular behavior, we are working the micrometer length scale or millimeter length scale, which is the length scale relevant for cells. So one cell is about the diameter of our hair, to give a reference.
And so I think this leads to the next question again. What kind of tools does your lab use to conduct this research? And since you're working on multiple scales, and why are these tools necessary for this type of research?
The main tools that we use are force measuring techniques. So we perform a lot of force measurements at different lengths, of course, based on the questions and the system itself. So we work with an apparatus called the surface forces apparatus, which is the grandfather of the atomic force microscope, the AFM. And as I was mentioning or explaining previously in the context of length scales, we work with large surfaces. Of course, we control the geometry of these surfaces, and we control how smooth these surfaces are as well. And we bring them together and we separate them at controlled rates using different motors and using actuators. And with that, we measure the interaction forces between the two bodies that we bring towards each other or that we separate. To measure adhesion, for example, we measure, of course, how much force we require to separate the two surfaces. If we are measuring friction, like for biotribology, we deposit natural lubricants, like synovial fluid, for example. We confine and then we displace the surfaces relative to each other and measure how much force we need to apply to these bodies to make them slide against each other. So that's for biotribology. So we use the SFA, the surface forces apparatus, and that gives us extremely high force sensitivity. So we are measuring forces at the millimeter, millinewton, or fractions of millinewtons. Now remember, 1 newton is given by, or 1 kilogram is about 10 newtons. For the cell-related work, we use also platforms that allows us to measure the forces that the cells generate. So for that, we combine optical and fluorescent microscopy. So we need to see the cells somehow. So we use a microscope to see the cells. And we work with a technique called traction force microscopy, in which you have elastic or compliant substrates. So substrates that can be easily deformed by the cells. And similar to your background, Edgar. So we have a lot of fiducial markers, which are small beads with which we embed in the substrates, and we can see with the microscope the fluorescence. And we have a cloud of fiducial markers. And when the cells deform the substrate, the fiducial markers, or the beads, are going to be changing position. So we can track the position of the beads at any given instant. And by monitoring the change in position, we can know how much force the cells are exerting on the substrate, applying to the substrate. So that is the measuring technique. And of course, we can come up with different perturbations. We expose the cells to smoke, to drugs, to medicines, to enzymes, to see how they respond to what we call a perturbation. Okay, does that make sense?
Yeah.
I mean, of course,
I guess
all these things without graphics, without sketches, is yeah, it's not that simple.
Well, that's what I think I was going to bring that up is, can you explain a little bit what fluorescence is for those that don't know, and exactly how you choose the fluorescence per cell? Is there a specific methodology that you use to say, oh, this fluorescence is going to be used for this specific work? And for those that don't know what fluorescence or fluorescein is, can you explain a little bit on that, so that even though we don't have a visual representation, they understand like, oh, that's what that is?
That's a very good suggestion, Edgar. Fluorescence is the phenomena in which you excite a molecule with a light, for example, a visible light. Let's stay with visible light, and the molecule will emit photons with another color, and then you can see that. Okay. We have a natural fluorescence in the oceans, for example, phosphorus, fluorescein, as you mentioned. And the fluorescence that we use, we purchase materials that are already designed to emit or to fluoresce at certain wavelengths. So we can purchase beads that are red, or beads that are green, or beads that are yellow. And when we embed them in our substrates, we excite them with a light, but we know they're going to be emitting. So the light that we use is it's black. It's an halogen lamp. It's really a broad spectrum source. And we use filters that are going to collect only yellow light, or only green light, or only at the wavelength that the selected particles are going to be emitting light.
Got it. So you use light at some intensity or wavelength, or you use the fluorescence at some intensity and wavelength that it absorbs some stuff and it emits the light at another color. Then you use a filter on a microscope or an apparatus that lets you see everything from that color, essentially.
That is correct. That's a very good summary.
So I know you have students working in your lab, and I'm not sure how many you have. So for those that don't know, where's your lab located? How many students do you have, both undergrad, grads, and postdocs?
So the lab is currently built by one postdoctoral scholar, the Jean, a graduate student, Ariel, and several undergraduate researchers: Joyce, Isha, Tiffany, Kate, Sai, and Mohammed. And they come from different backgrounds. So that's also very nice. We have, of course, researchers from any stage. So something that I really like is to have the lab open for everybody, as long as they're interested and curious at different levels, because everybody can contribute differently always. But they're all coming with different backgrounds: bioengineering, material science, chemistry. We have biologists in the lab, mechanical engineers, etc.
Okay, and so your lab seems to be very diverse, and it sounds to have a few students right now. So are you currently looking to expand your lab? And if so, are you looking for a new grad student, postdocs, or undergrads? And what kind of work are you pursuing if you're trying to get new students? because I know you have a lot already, so I'm assuming there's all kinds of projects going on. And so
there's always something else to do. So we are not short on ideas, no? And if students, regardless of the stage, no, undergraduate, graduate, postdocs, I think the most important thing is to hear if someone is motivated and curious about the type of work that we're doing, please reach out to us, and we can always discuss different projects. And of course, depending on what experiences, skill sets they have, and what they want to learn, we can tailor the different projects for the particular researchers. So I think that the only requirement is to be curiosity driven, motivated, and have fun in the lab.
Actually, yeah, that just answered my next question, which is almost answered my next question. Why should someone consider joining your lab? Whether they're undergrads, grads, or somebody that's looking for a postdoc position. And I think you just answered it. And I think it has to do with, like you said, the being curiosity driven, right? And being just motivated to do the type of work you're doing, it seems, right? That's the student you're kind of looking for.
That's it. And of course, understanding that what we do is mostly for spectroscopy. And if that's something that triggers interest, and of course, in particular systems right now, but we're expanding collaborations. But if someone is interested in really becoming an expert in synthesis of particular polymers, this is not the place. I don't have that expertise. But we can seek collaborations.
Okay, so you're pretty much open to saying like, oh, a student kind of comes into your office and wants to just kind of pitch an idea. That's kind of the thing that you kind of are open for, as long as they have some sort of like backup documentation, some papers, and if your lab coincides with that, right?
Yeah.
Okay. Now I think more about your lab. We want to hear a little bit more. I think people want to hear like student life obviously is important, right? As grad students, we tend to have a lot on our plate, even when we shouldn't. So research is a tough thing. And how do you keep your group motivated in conducting the research? And more specifically, something like cells that require maintenance around the clock. How do your students cope with this? And what do you do as an advisor to help these students, stay pumped up about the research and want to continue it?
How exactly the students do it, I don't know. I cannot speak for them, but I can tell you what we do as a group. One is, of course, to keep the interactions as frequent as possible and also spontaneous. Like, let's interact as much as we can and chat. At the end of the day, a healthy group, to my eyes, is formed by healthy individuals and interacting regularly, inside and outside. So we go for lunch regularly, we do activities outside of campus, and we do activities also internally. And we approach each other. No, I think trying to have really an horizontal and respectful relationship is crucial. And I already answered to some extent this question. Like, if the people that approach me are curiosity driven, motivated, and they find the questions that we have relevant, and I think the motivation is already there. No, it's just executing the work and really start to answer. No? Like that, at the end of the day, they are here because they want to find answers to these bioaddition, biotribology, or epithelial mechanobiology questions, and that's it.
All right, so that actually sounds great, actually. And so I guess the next question is, aside from the self-motivation and whatnot, you have a lot of tools, right? So do you provide training for these students that start at your lab, especially thinking about undergraduate researchers, even graduate and postdocs that may have never used some sort of apparatus like the ones that you use for adhesion or force? Do you provide that kind of training yourself, or do you have already a guide that kind of says, oh, here are the steps to using it as simply as possible. Try this example and then kind of go on your own.
No, I think I'm going to try to keep it like this for as long as possible. No, I really like to spend time in the lab, but I cannot spend a lot of time in the lab. But I'm so far being able to train everybody in the particular experimental tools that we have. I wanted to connect it to the previous answer. Now that the group is slowly growing, of course, there's more knowledge already in the lab, and we can train each other on the different, not only the characterization tools that we have. It is about the approach. How are you thinking about the problems? For me, it's very important that everybody understands the details of each, the fundamentals, the physical principles under which each of the techniques are operating, so that we can really understand the data, troubleshooting, experiments, troubleshooting or fixing the equipment. And I'm going to be, so at the end of the day, my job is to be a mentor and help and train and teach everybody to use and conduct research in the lab. But I'm just a mentor, no? At the end of the day, it's all of you or all of the researchers that are going to be really the ones putting the time, and that's going to be depending on each of them. So I'm very, again, I really believe in the intrinsic motivation of the people that are joining the lab, and of course I'm expecting initiative. But they can be sure that they will find support in me.
So then I'm assuming that there's some students that you've had that then end up kind of, in a sense, not literally, but surpassing the skills the skill sets that they end up coming in with to use these tools, right? You end up teaching them some basics, and then next thing you know, they're doing different either methodologies or manners in using these tools that further their skills beyond what they even thought is possible, right? You allow them to kind of explore further beyond that.
I hope they can do that. No, at the end of the day, the laboratory is a creative space. No, I call it a creative space or a creative playground, and it is a unique opportunity to have that space and explore with the materials, explore with the techniques, just explore. Of course, consciously, we need to talk about their limits. Of course, safety is the first thing that we need to keep in mind. No, there's some boundaries, and as long as we have those boundaries clear, it is a playground that should allow everybody to push the potential.
Got it. So that's something I think a lot of people look for in labs. I mean, I specifically love the freedom in being in a lab and just being able to do whatever training I want to do, even if it doesn't necessarily align with my research. So I think students hearing this will definitely take that in good conscience, because it's I don't think it's something that everybody advertises, and it's something definitely important, especially to those people that don't know how to use these tools and that have an idea and don't know how to approach it. So thank you for that. And this is probably going to be our last question, and it's, I think, the easiest one. Aside from all of this research, aside from all of the teaching and everything that you do, what other interests, I guess, even within academia or outside, do you pursue that make you stand out within your I guess either your department or just in general in your research field?
The technique, so outside or inside the research field?
It wouldn't matter, either or.
So one of the things that, for example, techniques in the lab that are unique is the surface forces apparatus, this force spectroscopy technique that I described previously. There are very few around the world. So we have the capacity to measure different types of systems at the molecular scale with unparalleled precision and with access to interfaces. So gaining that skill set will be unique. There are about 30, maybe 40 laboratories with this technique worldwide. And I was exposed to this technique during my grad studies, and I fell in love with the technique. It's extremely powerful. And some of the other techniques that we use, they're very well known and established and used in many laboratories, so we complement with that. And outside of the lab, I encourage everybody to travel as much as possible. That has been eye-opening for me. I've had the opportunity to travel a lot, and I've learned a lot by doing so. Not only but for the fact of traveling, it's the fact of getting exposed to other people, to other cultures, to other ideas. And I think that's crucial. And it doesn't have to be traveling to the other side of the world. Just going outside to the mountains that we have here so close by in Yosemite, or to the Bay Area, to Sacramento, to Florida. Just get out and get to know as much as you can from your surroundings and interact with people.
Yeah, and I think that's important, because definitely even in my research, it's one thing that I realized, that if you are exposed to more, whether it's just nature or research articles or conferences, there's always something to learn when you do more things outside of the lab itself, right? And so that's definitely something important to point out to everybody. I think that was the last question I had. Is there anything else you'd like to let students know about your lab, about any of the research that you are conducting? I'm happy to share some information that you like to provide, and so that students, can see more of your lab.
Yeah, the lab is always open. If you're curious to see the techniques that I described today, reach out, and I'm happy to give you a tour and explain the techniques and explore possibilities. But more than that, I want everybody to encourage the undergraduates, particularly, to explore research opportunities. It don't have to be with me, of course. Just approach any professor on campus, or wherever you are, and if you are interested in or curious about the research that they're doing, going and doing research in the lab. And of course, it can be experimental, it can be computational, it can be theoretical, it can be a combination. It is an eye-opening experience. So don't hesitate to approach to whatever professor you are thinking about approaching to seek opportunities.
That is a great way to end our interview, I think. I don't think I have any more to top that off. And I wanted to just say thank you for pretty much joining me on this interview today, Roberto, and being able to talk about your research and encouraging students to continue doing so. And I hope to interview you again to talk more about your research later on, and continue this talk with other professors as well, so that they're able to not just, again, talk about the research, but also talk about how the student life within their lab is, and encourage others to also pursue more higher education on not only, as you said, your field, but other fields as well. So on behalf of GSA and myself, thank you for joining our podcast episode for today.
Thank you for the invitation, Edgar. It's been a pleasure.